Okay, welcome everybody to the March virtual lab meeting for Hao Fountain Syndrome. My name is Amber Freed and I'm Executive Director, and we have founder Bo Bigelow and President Becky Ratz on the phone. Bo has a fantastic podcast. Bo, if you're in a place, I saw that your announcement for your podcast just quickly this morning. Okay. Typically he takes this call when he's dropping Tess off to school, so he is not in a place to speak. Is anybody new here and would they like to introduce themselves?
Can you all hear me?
Yes. Hi, Alexis.
Hi. So my name is Alexis. I am a genetic counselor here in Detroit at the Children's Hospital, Michigan. We recently identified a patient on genome who has a really suspicious USP7 variant that's actually been described in literature before. We don't have a lot of family history for him, so we have been trying to kind of start from scratch and figure this out. He does also have an affected sister who is getting testing soon, so might be someone else to add to the registry. Him and his mom are part of the registry right now.
Oh my gosh, that's amazing. And you are the best genetic counselor in the world. Thank you for advocating for your patients and being here today. And you have the world expert even on the phone. There is Dr. Schaff in Germany.
Yes, it's nice to meet you, Dr. Shaw.
Hi, it's nice to meet you, Alexis.
What a fabulous call. And this works out perfectly for some other things we're going to discuss today. We do have a roster here. I'll put the link in the chat. Update your information if you'd like or correct it. There are some funding opportunities right now through the American Epilepsy Society and Simon's Foundation. Going on our 2026 goals, Alexis and Dr. Schaff, we have so many clinical goals this year. We're going to publish our disease concept study, establish a strategic research plan, and what we really need help with is our standard of care. We are recruiting clinicians and other healthcare workers for USP7 right now. If you know of anybody, we're about to send out a large recruiting email and just start. I'm not sure. We hope that this will be a good foundational start to something we can build on. We're also looking at analyzing our data in matrix further, fostering centers of excellence, and ideally we'd like to put in our LOI to have an FDA listening session this year. We are determined to make future drug development much easier. And then scientifically, and this is a good time to mention, I have been totally up to date on my emails. I've been very proud of myself for that. And last night I realized all of my USP7 emails had been stuck in an outbox that just decided not to send and receive for a couple of weeks. Dr. Fontaser, I sent the reply you got yesterday weeks ago. So I was even surprised when you emailed me back. I was like, oh, this was a while ago. So we were working on a small molecule screen along with Charles River, and one big conclusion that came from beginning that screen is that we need to understand the basic science more. We need to understand exactly what we're targeting and what we should be measuring to know if we are activating the gene. So what this means is more basic science, and we have a really fantastic core group of scientists. And what we would like to do is involve the people actually doing the work into our strategic research plan. Develop the questions of what do we need to answer with the end goal of a small molecule screen. Any questions about that? It's still high level. There will be more to come.
Well, maybe, if I may, Amber, I would have a question about the clinical goals. To publish a standard of care, which I think would be wonderful. But when we met at the family conference in Nashville last year, we realized, or we felt, the group felt that there was really no clinical champion for Hao Fountain Syndrome in the US right now. Like, are you guys in touch with anyone who would kind of fill that role?
It's been a problem. We have some endocrinologists that are very interested. We're weak on neurologists. Our community is so spread out
Hmm.
across the US. It's been really difficult, and we do seem to have a cluster of patients that go to CHOP, Children's Hospital of Philadelphia, but they're some of the most difficult people in terms of bandwidth.
Hmm. Yeah. Because I think that that would be key. You know, we have, I think, in order to publish a standard of care, like we do have a starting base, and like, Sebastian Seiler and I wrote the Gene Reviews article, which does have a section on kind of, clinical assessment and management. But obviously, the focus of the Gene Reviews is more kind of on the genetics and genotype, phenotype stuff. But it does have bits and pieces that could be used as a starting point, but it would be so meaningful to connect with other clinicians who have seen, let's say, more than three individuals with Howe Fountain syndrome to kind of really put something meaningful together.
Yes, absolutely. Any good ideas on how to? We're asking our patients, any other good ideas of how we can search for those people through our patients, but also from a medical community standpoint?
I don't know. I think starting with the patients and kind of inquiring with them, like who have you seen, who has really stepped up to kind of provide exceptional care and kind of, care deeply about your child, yourself, and the related condition would be a good starting point. And if there were names that were mentioned kind of multiple times, those may be the people to connect with. Yeah, I don't know if like Teresa or Mark, from an FPWR side, kind of if you have other kind of suggestions.
I mean, we could, so we have a clinical network of PWS physicians, and we send them communications sometimes. We could, you know, sort of highlight that there is a need for clinicians to care for people with Howe Fountain. Um, but I think, yeah, I think that the families identifying physicians is probably the best way to go because that way there's the personal connection and you're likely to find someone who, is sort of really interested and has at least a personal history of stepping up to care for individuals. But we'd be happy to sort of include something in the next, we send something out every few months to this click database. And actually, yeah, we've just started like a listserv, so we could maybe sort of just query, if anybody knows of someone who would like to become more involved.
Yeah.
We could do that.
And and we should connect with the French group because, given that France is so centralized, it could well be that many of the French patients, and they are very well organized, Manger la Vie, so it could well be that multiple of them kind of go to Paris to a certain physician kind of taking care of them. So we should definitely inquire with them.
Okay.
And I can tell you, I reached out to the Houston families because we have a cluster of families in Houston and they all go to somebody different. It's all within the same system, but they all go to different clinics.
Yeah, that's unfortunate.
Yeah.
Yes. So, uh, sorry. Okay, so then we have so many resources. The biobank, we have a resource spreadsheet of mice, we have data through Matrix. We're having our symposium in Philly, and there will be much more to come on that. It will be August 7th. And lastly, why everybody is here, we are going to be talking with Cyogen about supporting drug development for Hao Fountain and what they do for disease models. So with that, I will hand things over to Ben. We'll have some discussion at the end and I'll be back. Hold on, let me
Yeah,
stop
I'm
sharing.
muted. Okay, I was muted. Okay, so thank you, Amber, for your kind introduction. So this is Benjamin Chu from Cydrin Biosciences. So actually, I apologize. I'm the only one online today because Ivan and Matt are actually attending the Next Generation Biomed conference in London right now, and which is very important. And I'm not going to that meeting, so I was requested to present here. So thanks Amber again, because Amber is our old friend. We start connecting from this SLC6 and 1A1 community, and then actually we were introduced to Becky and Bo, and that connect us together here. So today, in the next, let's say 25 to 30 minutes, I will share you which actually models and services we have from actually a provider side of view, and then followed by a very proactive discussion, I believe. So what we can help you and empower your research, drug discovery, and etc. So please allow me to share my screen. So can everyone see that? Yes, just let me move actually our window somewhere. Okay, so yeah, so you can see actually my talk. So I'm Ben Q. I'm Business Development Manager for Southern Europe and Scandinavia. Good morning and afternoon, everyone. So today, actually, my title of speech is Supporting Drug Development for Rare Diseases and Exploring Support for the Foundation of Hao Fountain Syndrome with Cydrin's integrated in silico, in vitro, in vivo models, as well as CRO platforms. So actually, we are Cydrin, so a 20-year company headquartered in Santa Clara, USA, California. So we have over 1,000 employees distributed in the head office, but also branch office in the Rotterdam, the Netherlands here, Japan, and also 3 key facilities in China with animal facilities. So our animal facility was accredited by ALAC and also assured by OLO. We actually are an animal provider, but also a CRO service company. So starting from 20 years ago, the key technology was genetically modification in the embryonic stem cell ESC. And then actually Cydrin extended this technique to rodent models, including mice and rats, also the iPSCs. And around 5 years ago, due to the COVID pandemic, so we closed the door a bit and we established our CRO platform. So we have 5-6 big CRO platforms, and making use of the advanced models, including Hugo GT, we will hear in the following slides, and Hugo App antibody mice. And Cydrin was able to collaborate with actually over 10,000 friends and with over 10,000 citations, so co-publications as well. So we were involved in over 58,000 projects, and currently we have over 800 available models. If you look at what we can do, so this slide shows you our spectrum of models and services. So you can clearly see in this summary, so we are focusing on preclinical studies, starting from target identification to lead optimization, in vitro and in vivo analysis, but also CMC and the IND, so the drug declaration. And very recently, we actually extend our business to this clinical phase by actually starting the CDMO or GLP level laboratories. And one thing I have to stress is like many years ago, our CEO Lance was very interested in rare diseases, and also based on his actually background, so as a computer actually student and fellow, so he actually already thought to recruit AI into the system and to empower our portfolios. And therefore, actually, so these slides, I want to show you how to actually study a rare disease and develop drug candidates. So the translational funnel as shown on the left side. So usually we have some clinical information, including human genomic validation. And to follow that, we make use of precision genetic modeling, including in vitro tools, cell lines, iPSCs, and in vivo animal models, mice, rats, or primates. And in the end, that leads to actually IND-ready integrated package. And today, in my talk, I will tell you a few things: the rare disease database, the genetic engineer technologies in brief, and iPSC in vitro models, and Hugo GT as a significant in vivo model, as well as a few CRO demo case. So the first thing is, what is the RDDC? So RDDC refers to Rare Disease Data Center. So this is actually the main view of the website. It's completely free for public. One of the major advances of the RDDC is it's a very nice and comprehensive summary that connects genes, diseases, and mutations, or I would say variants from patient information. So this database contains over 63,000 gene entries from different species, covering over 21,000 diseases. And AI helped to summarize, but also rank, sort a bit from the other platforms and literatures, the platforms containing ClinVar, Maracas, or OrphanNet, it's for rare disease definitely. And the good thing is we also extend that genes and diseases and correlate them with the mutations. So we actually, this database has over, you can see 2 million mutation information coupled to these genes and disease. Here shows you just the example I have done in the afternoon in local time Europe. So I have searched USPC gene in the RDDC database, and on the left side the corner you can see the disease database. The USPC gene is actually responsible, of course, for Hao Fountain Syndrome or Fountain Syndrome. So AD refers to autosomal dominant and AR for autosomal recessive. But also I want to say the USPC gene is also involved, not a major gene, in some other rare disease such as this Schaff-Yang syndrome. At this corner you can see actually the available models, likely so far 6 due to the undevelopmental status. So you can see Cedrin's models. You can also see the models available from Jackson Laboratories. I assume they are knockout or flux models with a few publications available. On the right side, you can see actually the mutations or variants selected and ranked by the AI. So these are actually the mutations, no sense mutations, all the mutations coming from patient information. So I want to say is using this database, you can not only study the USP3 gene, but also other genes involved in rare diseases. So second part of my speech is Cedrin's gene editing technologies. Due to the time reason, I can't tell you more details regarding one particular technologies, but rather than an overview of a combinational toolkit of our cutting-edge model generation technology. So this slides for in vivo models. So we have 100% IP right belonging to Cedrin, this turbo knockout gene editing that can shorten the turnaround time from a year to 6 to 8 months. And there are ESL gene targeted gene editing, and CRISPR-Cas9 mediated homologous directed repair, as well as this regular transgenesis, for example, transgenic mice. We also recruit this piggyback transposon and bacterial artificial chromosome platforms to empower this in vivo model generation studies. And coming to the in vitro part, so as I said, we have induced pluripotent stem cell or iPSCs available, where I will show you more details in the following slides. But we also have the capacity to generate a normal knockout, immortalized cell line, knock-in cell line carrying a point mutation or point mutations, but also overexpression and knockdown cell lines as well. As actually an very fancy, popular, and advanced model, in vitro model. So I will show you some information regarding the iPSCs that are highly actually involved in, for example, neurobiology studies. So Cedrin is able to reprogram fibroblast or human PBMCs into iPSCs. And on top of the iPSCs, we are able to introduce mutations, so gene editing. And after this editing, we are able to differentiate these iPSCs to a certain type with a terminant phenotype. And this can be found in the textbook. So regarding iPSC differentiation into 3 directions, we have actually this ectoderm, mesodome, and endodome roots. So for your interest, I think most of actually the neuron specific cells are coming from this ectoderm, and this is an example. So Cedrin is able to provide you different types of these neuron cells, including these neuron progenitor cells, for example, cortical neuron and DOMA neuron, and also GABA neurons. And I will show you one example due to time reason that is actually Cedrin's iPSC-derived motor neuron. So this is showing you a standard 4-week differentiation process using our own protocol. So we start the starting material is iPSC, and actually the terminant product is actually the motor neuron. And what you can see on the top, the morphology, and we stain the cells at day 12, so in the middle of the differentiation, but also day 28 by the end of the differentiation with a matured phenotype. We actually introduced these neural markers 2J1, so you can see actually clearly at day 28 post-differentiation, so the cells shows this neuron, motor neuron phenotype in morphology. And on the right side, you can see actually we measured this voltage-gated sodium and also potassium actually voltage, but also on the top, the evoked and spontaneous action potentials. Actually, more significantly, so we actually Cygene is able to modify, as I have said, the iPSCs prior to the differentiation. So this is actually a very nice example to showing you a suitable model for amyotrophic lateral sclerosis or ALS. This is not a rare disease, but it is definitely a severe disease. What we have done is we have found actually one of the mutations frequently found in the one of the pathogenic genes, TARDBP or TDP43. It's M337V mutation, and we were able to actually genetically modify the iPSC to carry that mutation, and then we actually differentiated the cells to the motor neuron, and as shown here at the corner. So at the bottom, this is actually the mutant compared to the wild type on the top. So you can see clearly we still use this 2J1 staining compared to wild type cells with very clear and nice motor neuron morphology. And the mutant M337 showed actually a bite actually maturation, but also actually as well as the thinner, for example, membrane. Actually, then if we used patch crab electrophysiological measure to determine the actually membrane voltage potential, we can see for the mutant highlighted in red column compared to the wild type in blue, they showed actually reduced this membrane potential as expected. And this model can definitely be used for the further drug screening, small molecules. Currently, actually, we have summarized the available iPSCs with the mutations we have tested. So what you can see is these mutations in the iPSCs available for example, Alzheimer's disease, AD or ALS. But we would be more than happy to try to know actually the patient-derived mutation from you in this USPC-mediated Hao Fountain Syndrome, and we actually can engineer this and introduce this mutation into the iPSC and differentiate the cells for you. So next, I will talk about one of our advanced in vivo models, the Hugo GT. This slide actually summarizes the current methods to generate, let's say, humanized mice. So in the very old days, the transgenic was the traditional method. However, after a few generations, they show actually some unstable phenotype you have to match, and you have to mate and do make this further screening. And then over the years, so researchers started to use this point mutation of this CDIs, the partial humanization, as shown here. So with just assemble the human coding region and put it in front of this mouse gene with a stop codon. So under the promoter, this human gene can be expressed, but not the mouse gene. And currently, we believe the most physiological model would be our Hugo GT. So what you can see the difference is we replace in situ the entire mouse gene with this human ortholog. It's with everything. And on top of the Y-type Hugo GT model, we can actually introduce point mutations. So far, over the years, we have accumulated 204 available Hugo GT models. You can find all of them on our website. The Hugo GT refers to humanized genomic ortholog for gene therapy. They come with numerous benefits, including first, they were designed for preclinical evaluation of clinical drugs, particularly including siRNA, CRISPR-R, and antisense oligonucleotide, because these models ensure full-length coverage of pathogenic genes, not only including extrons, but also introns and 3' UTR regions that are frequently the target of siRNA and ASO. And they are also customizable based on individual needs, because our techniques allows to knock in up to 350 kilo base pair, so allowing this method of generating a human GT model available. And we actually divided or clarified our models into different subsets. So what you can see is on the very left side, in the neuroscience and muscle. So we have actually models for Alzheimer, for Parkinson, for Rett syndrome, but also for example, spinal muscular atrophy, SMN. I will talk as an example. So how we further develop and evaluate this model to meet actually the basic research and drug discovery requirements. Coming to the case study, so the spinal muscular atrophy SMA. So this is actually a rare disease, definitely a rare disease, and it's autosomal recessive neuromuscular degenerative disease. So in humans, there are two genes, SMN1 and SMN2. The SMN1 gene is able to produce 100% functional SMN protein, and the SMN2 carrying a point mutation in the exome number 7, C2T, only can produce 10% of functional protein compared to SMN1. And therefore, in patients, in the pathological scenario, patients like a functional SMN1, so the protein produced from SMN2 was not able to compensate the full function, and then patients become sick. And luckily nowadays, so it's possible to control, or I will not say cure, but control the disease by applying this antisense nucleotide ASO. However, in mice, the situation is completely different. Mouse only has one SMN1 gene. So therefore, in the old days, so I think from Charles River or Jackson's labs, so the method was they have to generate 3 mouse lines. The first one is the mouse SMN1 knockout, and the second is a transgenic human SMN2, and the third is the transgenic SMN2 liking the exon 7. So then actually they made these 3 mice lines together and generate the final mouse line for study. But the whole process, as expected, is complex. What we have done, we have this Hugo GT system. So we directly in situ replace the mouse SMN1 gene, because mouse doesn't have SMN2, with the human SMN2 gene. So we believe this replacement, a simple replacement, can already resemble and represent this disease scenario. And so we have the model called B6 human SMN2 Hugo GT model. The first information I want to share with you is, so please look at the survival rate. And for the wild type and heterozygous human SMN2 Hugo GT model, they are super they are actually variable. However, for the homozygous SMN2 mice, they actually start to develop severe disease, and they died from as early as, I would say, 10 to 15 days, and all of them are dead at 35 days. And from actually the feature, you can see actually the homozygous human SMN2 mice show severe muscle atrophy, ataxia, as well as actually the edema of the limb. Then actually histochemistry studies showed actually in this homozygous human SMN2 mice, the lymphocytic infiltration into actually the muscles compared to the wild type control. If we look at actually the expression level of the SMN2 in this model, so this is actually a Western blot on the top showing that in the homozygous mice, actually compared to the heterozygous mice, the reduced expression level of human SMN. And we actually tested one of the antisense oligonucleotide with a brand name Spinraza that are actually already on the market, and the drug name is ASO 1027. So we compare actually two injection minors ICV versus subcutaneous injections, and we can see actually both infection can actually elevate the expression level of SMN2. That's actually a rescue. And ICV through actually the brain spinal roots was actually better. We can also see actually this ASO treatment increased number of motor neutrons confirmed by this immunohistochemistry analysis compared to the vehicle treated group. And significantly, if this treatment actually was very good, and it can elongate the survival days of mice in this model. So what you can see is, as I have shown, in the untreated group, so all mice died at around 35 days. However, actually upon this ASO treatment, the survival rate was higher, and actually the survival days can be elongated up to 140 days. That actually makes this our humanized Hugo SMN2 model a superior model, and this we also compare that with actually the traditional model, the triple transgenic models, and they have actually very close pathological phenotypes and also a similar actually experimental window for the researchers. And to the end of my speech, so the last session would be our CRO services. So this slide summarize actually the available behavioral testings in our state-of-the-art CRO platforms, particularly for neurological studies, and we use different colors to differentiate the different diseases, including Alzheimer, Parkinson, Huntington, Rett syndrome, ALS, and pain. And you can see actually they are very traditional behavioral testings for motor or cognitive functions, as well as social behaviors, including the Rota-Rot, open field object testings, Y-maze testings, and Morris-Walker maze testings. And the CRO platform is actually designed for integrated neurological studies. So we have actually available models and how to use the models. So we actually can evaluate some drug candidates, and we use in vivo and ex vivo methods to this readout to evaluate the efficacy and safety of these therapeutic agents. And for the ex vivo studies, we have multiple molecular and pathological methods at the genomic level, protein level, and also these pathological studies. I just want to show you for the neurological studies, we are able to do these immunofluorescence stainings. This is an example in astrocytes. Also, the immunohistochemistry staining in microglia using these mouse brain tissues. We have fancy microscopes. And as a case demo, I want to show you a virtual example. For example, if we have one transgenic model, all humanized Hugo GT model, so we had a name, so with hyperactivity. So what we can do, so the first step is we actually start experimental arms, and we involve and employ these in vivo studies to look at the behavioral tests. For example, this open field test on the left side or the Y-maze test on the right side. So you can see clearly compared to the wild-type control mice, the transgenic mice on the left showed actually much faster running in the region, or they are actually very stressful to find functions compared to the wild-type mice. So that actually correlates with our aim of study to determine the motor or cognitive functions. And we are also able to further test these in vivo markers using, for example, electrophysiology methods. From here, you can see the EEG curve compared to the wild-type control. The transgenic model shows super active and frequent neuronal activity, the red compared to the green. And then actually we sacrifice the mice at the end of the test, and we use ex vivo markers, for example, special staining in the Golgi cox, or we actually, if we include the drug, small molecules, or small nucleoids, we can actually evaluate the efficacy of them. Since you have actually talked FDA, I just, in my last minutes before the presentation, I put this slide back. So you can see actually we have empowered multiple biotechs and farmers, for example, this Innovec, so to study rare disease, this XLRS and AMD. Well, very last, so if we go back and look at our capacities, so you can see due to time reason, my talk is focusing on neurosciences, but we also have the other platforms for immunology, metabolic disease, etc. I hope I have persuaded you. So Cedrin is your decent partner in preclinical studies and IND discovery. Thank you very much for your attention, and I'm very happy to answer your questions. Yeah, feel free to ask.
I would just be curious to the community if there's anything like need that you could utilize a model like this for now. We're always trying to think about how to prioritize like our capital spending budget.
Yes. So if we talk about actually new models, so if my memory is correct, please correct me if I'm wrong. So for this USPC syndrome, so the only available models are knockout and perhaps flox mice. So there are two actually directions. The first, you can find actually some interesting variants from patients and you analyze. So actually you perform actually a pie chart. So which mutation account for the most in patients, and we can introduce that point mutation into mice. So this is not humanized at all. But for some rare disease, that models can already represent the pathological scenarios. And the further step would be we can replace the mouse gene with the human gene, no matter how long it is. So we can actually we were able to knock in up to 1 million base pair. So definitely covers the whole gene that can replace in situ the mouse UPS7. And this would take, I would say, 6 to 8 months. But then on top of that, you can introduce this human mutation that takes another 6 to 8 months. But in this case, actually, this is the human mutations in the human gene, but in the mouse format. And for the in vitro studies, of course, you can use cell lines, you can use iPSCs. So this turnaround time would be much shorter. We can likely provide this cell line in 2-3 months, so 8 to 16 weeks to support your study.
Yeah, I mean, I think one of the challenges of Hao Fountain syndrome and USP7 is that there really aren't any major recurrent mutations. So the vast majority of cases that we have are just kind of individual, kind of personal missense variants. I mean, we have all different kinds of variants, like, nonsense missense, frame shifting indels, start codon, deletions, but as compared to many other conditions, including Schaaf-Yang syndrome and others, there really isn't a hotspot mutation, which makes it, somewhat harder to design a really meaningful and impactful model that will affect and apply to many of the patients. But one may consider kind of choosing certain variants in certain domains of the protein, because some preliminary work has been done showing that there may be genotype-phenotype correlations, and in particular, kind of variants in the catalytic domain of USP7 appear to be somewhat more severe compared to other variants in the gene. So that's certainly a discussion to have within the community.
Yeah, thank you, Christian. We believe maybe actually a pilot experiment using like cell lines, so to overexpress these CDIs, and we actually have some friends in the SLC6A1 field who actually use these modified cell lines. It's like HEC293 or I don't know. So to evaluate some actually neuron physiological markers of the protein, and you can compare a range of different mutations in the cell line format, and then actually you can pick up the one of interest or the one at this stage the most I would say significant or meaningful. Please, Boris, sorry to interrupt.
Yeah, sorry to interrupt. I'm gonna need to go. Just saying, like, to answer your question, Amber. Personally, we would benefit on having a mouse model for infection because we have viruses, we can infect mice, and because we know that on cells and in cells line, and we want to confirm in vivo, the infection is dependent on the USP7 activity. We have a readout for the function. So we can also test molecule to know if we can avoid having more virus, more infection, and it will like help us to have a view on like can we complement the function, only the gene, but the function. And I'm pretty sure that's the lab working in Boston and Connecticut on iPS cells and neuronal cells will benefit as well to confirm the phenotype they got in patient cells, for example, and having this IPS. And just saying, I contacted the French foundation right now. I sent a text to Jean Brun about looking for clinical doctors. He told me that each family is scattered in France, and they see their doctor like locally. They have there's some group in Paris, so I'm gonna ask them if they see the same doctor and go to the same clinic. They said the reference in France is this institution in Nantes, who does like genetic, but I'm not sure if they do only basic science or if they have doctor yet. So I'm gonna send them an email as well.
Has there been an approach to activate USB 7 with small molecule?
Yes, there has been a screen
Yeah,
here.
I was gonna say I can take that one because I did the work on it. We found a small molecule that binds to the allosteric site on the catalytic domain that copies the C-terminal tail and enhances the activity. But so far we were only able to do it for a truncated mutation in cells. And it was still like an engineered cell kind of assay, so it's still not anything concrete yet. And it's too weak, we would have to enhance its like potency.
What fold increase do you see?
When we looked at the one, no, I actually don't know. I can't tell you. We have the published paper for the small molecule activator of USB7 that talks about it like mechanistically, and then the functional spectrum of Hao Fountain Syndrome, which has a small part on the cell work, which is not the part I did. So off the top of my head, I can't tell you, but it's in there. They're both in PNAS.
Well, I think one of the best way, knowing the much broader complex role of USB7 and knowing the challenges of heterogeneity in the mutations or missense, nonsense deletion, I think the best approach would be for the foundation or for scientists to really screen for activators that would at least cover majority of the mutations. And it appears to me that that's where we should really focus, or at least draw interest in different labs as well as foundation and including CROs, because the assay for USB7 is pretty simple, and scientists have a habit of looking for inhibitors, not activators. So I think it's USB7 conditions can be established where you see very minor activity, and then you screen for small molecules that will activate the enzyme. And I think, that probably the best way for families, for patients, for clinicians. All other approaches are also okay. Gene therapy, adenovirus. But this is simple in terms of delivery and in terms of preclinical testing and safety and all that stuff. So that's my 2 cents. I think that nobody has discovered an activator of deubicrylases. Maybe, I don't know, USB 14, but that's controversial.
Yeah, there was another paper too by another group that also found activators that bound to the same spot that we did for USB7 as well. Which I can't remember. They looked at stuff that was already, I think, FDA approved or something like that. But that's also published. I just can't remember off the top of my head. So we all found the same mechanism of activation. So it really seems like a good activating spot, but it's probably also not strong enough currently to be used as is.
Well, yeah, I think assay is simple. Making enzyme is not hard. Everything obviously you published it, and so we need potent activators. We need potent activators, and it's possible. It is possible.
All right. Well, any other questions, comments? Thank you, everybody, for the great call today. I will send out a recording along with the notes and contact information. And this is a great place to continue the conversation and help the nonprofit prioritize our capital spending budget and the fastest way to start helping patients.
Thank you.
Thank you. Bye, everybody. Have a great week.
Thank you very much. Bye bye.
Bye.